So what is DNA replication? It is a process by which exact copy of parent DNA is made. Okay. So it is semi-conservative method because during each phase of DNA replication, one strand of the parent DNA is conserved. Okay. So suppose this is my DNA, and during replication each strand of the parent DNA remains and new strand is synthesized. So from one copy of DNA, total two copy of DNA are synthesized. In next, if we replicate again, then from 2, 4 DNA are synthesized. This is how DNA replication works. Okay. Now, what are the substance or proteins or enzymes that are required for the DNA replication? So it is DNA A protein, okay, DNA B protein, which is also called as a helicase, then single stranded binding protein that will prevent rejoining of the two strand of DNA. Helicase will cause the unwinding of the DNA. DNA A protein will identify the origin of replication. Okay. Then Topoisomerase, it will prevent supercoiling. Positive and negative supercoiling is prevented. We will see about it. Okay. Then main enzyme is DNA Polymerase and ligase and terminating protein. Plus DNA polymerase require the primer. Okay. So with the help of primase, primers are added. So all things and nucleotides are required for the DNA replication. So this DNA replication taking place in S phase. Okay. And in prokaryotes, it takes place in bidirectional. Okay. And there is a single origin of replication. While in case of eukaryotes, there are multiple origin of replication. Okay, and replication start at the 80 rich sequence. Okay, that is called as the ORI C site, where there is a multiple 80 rich sequence is there. So as we know, this is 80 is there are two hydrogen bonds as compared to CG. Okay, so it is difficult. There is a weak bond between A and T, so two strands of DNA can be easily separated. Okay. So again I revise DNA protein that will bind with the origin of replication, DNA helicase, which is also called as the DNA B that will unwind the DNA, primase that will add the primer, so that DNA polymerase will add the nucleotide. Single stranded binding will prevent the rejoining of the separated DNA strand of DNA. DNA topoisomerase will prevent supercoiling. DNA ligase will join the DNA molecule and enter binding protein. So there are three stages. One is initiation, elongation, and termination. So initiation, as we know, at the origin of replication, there is a starting of the DNA replication where DNA will unwind with the help of helicase and DNA A protein. Then with the help of DNA polymerase enzyme, DNA is added and new DNA strand is synthesized, and ultimately it is terminated. Okay. So there are three types of DNA polymerase in prokaryotes. That is called DNA polymerase I, DNA polymerase II, and DNA polymerase III. This DNA polymerase III is the main enzyme that will add nucleotide okay, in leading strand and lagging strand. Now what is this leading and lagging strand, that we will see in few minutes, okay. And this DNA polymerase read the nucleotide in 3' to 5', okay, and add the nucleotide in 5' to 3'. So always remember, nucleotides are added always in 5' to 3' position. Okay. Suppose this is a DNA strand, okay. There is a nitrogen base, ribose sugar, and phosphate, and this phosphate molecule. Here to this hydroxyl group, so this is a 5' end which is free, and to this side another nitrogen base, okay, another nucleotide is added, and here there is a phosphodiester linkage, okay. And so newly synthesized new nucleotide is added to the 3' hydroxyl group of the previous nucleotide. So always this synthesis takes place in 5' to 3' and read in 3' to 5', and add nucleotide in 5' to 3'. Always remember this, okay. So there is a replication bubble, and this ORI C site, which is 80 rich sequence, where this DNA A protein will bind, okay, and last then helicase enzyme will unwind the DNA strands, okay, which is also called as the DNA B. Now it can again join together, but it is prevented by the single stranded binding protein, okay, so unwinding with the help of helicase. Now when you unwind the DNA, you can see when you unwind the DNA, there is a chance that there is a supercoiling occur. What do you understand by supercoiling? That more twisting than the normal. If it is more, then it is called positive supercoiling, and towards the other side there is a less twist, which is called as a negative supercoiling. Okay. So this topoisomerase, what they will do? Suppose there is two strand of DNA, so it will nick at this side, and then it will make again reseal it, okay, topoisomerase 1, while topoisomerase 2 will cut at both the strand of the DNA, and so that it can again ligate. So two twisting of the DNA molecules are corrected, okay, and that's why how this supercoiling is prevented by the topoisomerase. This topoisomerase 2, also called as a guidase. So topoisomerase are the enzymes that will prevent the supercoiling of the DNA. If there is a supercoiling, then DNA polymerase cannot act. That's why this prevention of supercoiling is very important. Okay, you can see many videos regarding this topoisomerase action in YouTube also. So now we will see how DNA polymerase works. Okay. So you can see this DNA helicase that will unwind the DNA, single stranded binding protein that will prevent re-annealing. So we have seen that DNA polymerase III will add nucleotide in leading and lagging strand. Okay, it will add nucleotide in 5' to 3'. DNA polymerase II has a repair activity. Okay, and DNA polymerase I, it will remove the primer, okay, and join again, okay, join the remaining nucleotide. So it has an repair activity, and it has a removal primer because of its 5' to 3' exonuclease activity. Okay, so it will remove the primer and join it. And this DNA polymerase III has a 5' to 3' polymerase activity and 3' to 5' exonuclease activity, so that it can proofread. Okay. So if any mismatched nucleotide is added, it can remove with the help of 3' to 5'. So the DNA polymerase III is a very important enzyme. DNA polymerase I is for the removal of the primer. Okay, now this DNA polymerase work very efficiently, but it has one condition: it will add the nucleotide only at the 3' hydroxyl group. Okay, so here you can see there is no nucleotide. So how it will add? So here it requires one 10 to 15 nucleotide base, that is called as a primer. This primer is added with the help of primase enzyme, which is also called as a DNA G protein. So this primase is a, you can see, DNA dependent, okay, RNA polymerase, because it will add, this primer is nothing but short stretch of RNAs, okay, ribonucleic acid. So once primer is added, then only this DNA polymerase will work. Okay, and it will add the nucleotide in 5' to 3'. So you can see that once primer is added, now this DNA polymerase will start adding nucleotides. Okay, and as so towards the this is the replication fork. So towards the replication fork, the synthesis of nucleotide is continuous. So this strand where nucleotide synthesis is continuous is called as a leading strand. Okay. So DNA polymerase is very effective enzyme. It will add about 1000 nucleotides per second. It is very efficient, and it has a 3' to 5' exonuclease activity. So if any mismatched nucleotide is there, it will remove the nucleotide also. But it has a one condition: it cannot add nucleotides directly, so it requires the primer. Once the primer is added, so it's 3' and there is a hydroxyl group, nucleotide is added, and then this it is continuous. But what will happen to the other side? You can see here there is a primer is added with the help of primase enzyme DNA G, and nucleotide are added. But as DNA unwind, then what will happen? Suppose here okay, so towards this side there is a primer and nucleotide are added. As DNA again unwind, okay, towards this side, the lagging, this is called lagging style, because with the help of primer and nucleotides are added. But as again DNA unwind, then again it requires the primer, and it will add the nucleotide up to the last primer. Again DNA unwind, again primer is added with the help of primase, and it will add nucleotide up to the last primer. So here you can see that the nucleotide synthesis is not continuous, it is semi-continuous. So it is lagging behind the primary strand, leading strand. So it is called as a lagging strand. Okay, and these fragments, they are called as the Okazaki fragment. Okay, so you can see here as DNA unwind, every time it requires the primer. Okay, so if I draw the diagram again, this is a 5', this is a 3', this is a 3', this is a 5'. So here with the help of DNA G, primer is added, and nucleotide synthesis is continuous. But what will happen towards this side? Here, this is the 5' and 3' end. So primer is added, and nucleotide synthesis takes place. As DNA unwind, again primer is added, and it will add the nucleotide. See that here it will add the nucleotide up to the last primer. Again DNA unwind, and it will add the nucleotide. Okay. So that here towards the other side, away from the replication fork, DNA synthesis is discontinuous. That is called as the Okazaki fragment. Okay. So I hope it is clear to you. Okay. So you can see here the DNA synthesis towards the replication fork is continuous. Okay. So elongation here it is continuous. While in another strand, as the DNA is unwind, there is a nucleotide are added. Okay, so it is lagging behind. They are called Okazaki fragment. Okay, and after that, primer is removed by the DNA polymerase I. Okay, and then joined together. Okay, and that is by the DNA polymerase I. And nick is sealed, gap is filled by the DNA ligase. This is how DNA replication takes place. As in prokaryote there is a circular DNA, so once it reaches to the other side, then it will automatically replication is stopped. Okay. So now what are the inhibitor of DNA replication and summary? So conversion of DNA to DNA, it is a semi-conservative method. It is a bidirectional. Here DNA polymerase read the nucleotide in 3' to 5' and add the nucleotide in 5' to 3'. Primers are required, which is added by the primase enzyme, which is called DNA G. There is a leading strand and lagging strand. Three type of DNA polymerase I, which work as a proofreading, which remove the primer because of its additional 5' to 3' exonuclease activity. DNA polymerase II has a repair activity, while DNA polymerase III, which will add the nucleotide in leading as well as lagging strand. It has the DNA polymerase III has the 5' to 3' polymerase activity as well as 3' to 5' exonuclease activity. So any mismatched nucleotide is there, it can cut at the same time. Okay, and RNA primer is around 50 to 100 nucleotide, and prokaryotic DNA polymerase are 1, 2, and 3 portions. So we have seen that main is DNA polymerase 3. 1 is for the removal of primer and gap filling, and 2 is for the repair. Okay, there is a single origin in prokaryotic. It adds nucleotides 1000 to 2000 nucleotides. Okay, so only one condition that it requires a primer, and rate of replication is 500 nucleotides per second. Okay, so this is about the DNA replication in prokaryotes. There are some antibiotics that can remove the that can stop the DNA replication. So one of it is quinolone group, mainly the ciprofloxacin group. Okay, so you have heard about this group, ciprofloxacin. So ofloxacin, ciprofloxacin. Okay, they are called quinolone. So what they will do? They will inhibit topoisomerase enzyme. So what is the role of topoisomerase? This topoisomerase, they will prevent the supercoiling. So if we inhibit the topoisomerase, then there will be supercoiling will occur, and because of that DNA polymerase cannot work, or helicase cannot unwind the DNA, and that will lead to the inhibition of DNA replication in bacteria, and that's how the replication is stopped. So bacteria will die ultimately. So this is how this ciprofloxacin drug, they will inhibit the DNA replication. This topoisomerase has two parts, 1 and 2. 2 is called the gyrase. Topoisomerase 2 will remove the negative as well as positive supercoil. Okay, it will cut the DNA on both the strands, while topoisomerase 1 will cut the DNA at one strand and again join. So one twist is removed with the help of topoisomerase 1. Okay, so it will remove the negative supercoiling. Okay, that's how DNA replication works. I hope you understand uh, about the DNA replication. In my next video, I will discuss about the DNA replication in eukaryotes. Thanks for watching. If you like my video, please hit the like button. Please share my video. Please subscribe to my channel. And thank you for watching. Bye.